So as Valerie kindly introduced me, I am Elena Correa Puertas, and I'm a postdoctoral researcher at CentrEau Research Center in Montreal, and this is associated with Polytechnique Montreal. And today I will be talking to you about microplastics in the aquatic environment, and we will try to answer the question on how bad are they compared to the rest of the environmental impact of a product over its life cycle. À la fin, vous pouvez me poser des questions en français aussi, si vous voulez. So before we get started into microplastics, I will give you a brief introduction to life cycle assessment, since there's people from different backgrounds here and I want to have everyone up to speed. So just as a quick introduction, in life cycle assessment, we look into different life cycle stages of a product, all the way from the material extraction, the production, distribution, transportation, use, and then the product is disposed at the end of life. And for each of the life cycle stages, we look into the emissions as well as the resources that are used. So overall, we want to see the impacts over the entire life cycle. So how does that work? Well, we have the different emissions and resources, and in life cycle assessment, we link them to different impact categories. So for example, CO2 could be linked with climate change, and this ultimately can cause damage on different areas of protection, such as human health and ecosystem quality. Or another example, NOx can lead to the problem of acidification, which ultimately causes damage to ecosystem quality. So in life cycle assessment, we try to be as complete as possible, gathering all the emissions resources used. For example, resources could be water that lead to water use and eventually water scarcity, which can affect human health and ecosystem quality. So this is just a brief introduction into life cycle assessment and how do results of life cycle assessment look like. This is a case study that was done at Polytechnique Montreal in the food cafeteria for the students. And they were looking into different food containers. So there was one plastic option, which was expanded polystyrene, and two compostable alternatives. One was bagasse and the other one was cardboard, as well as a reusable ceramic plate. And on the left side, we see the ecosystem quality results. And actually we see that the plastic container has the lowest potential impacts on the environment, whereas the cardboard container has the highest potential impacts. And that for human health, similarly, actually expanded polystyrene container has among the lowest impacts, only lower by the reusable container. And then the bagasse containers have higher impacts on human health, potential impacts. So how is this possible? And we might ask ourselves, are plastic options better, although they're banned? But the problem here is that when this study was done, there was no methodology in life cycle assessment to assess the impacts of plastic emissions. So here we were looking into climate change, water use, and different emissions and resources among different life cycle stages, but not what plastic emissions cause. So this is why we need to take life cycle assessment one step further in our research in order to include plastic emissions, so in kilograms of plastic, and link them to different impact categories and ultimately to different areas of protection. So for example, we could use characterization factors, which are linking the plastics all the way to the damage they ultimately cause to ecosystem quality, and the unit would be a potentially disappeared fraction of species over a specific area and time. And now let's break this down into the steps and the research that has been done already. So for the first part in life cycle assessment, this part on the left, we call it the life cycle inventory. And here is where we gather all the inventory flows. So in the case of plastic is to gather what are the plastic emissions of a product. And then in the second part, there's the life cycle impact assessment. And this is where we link the plastic emissions with the potential impacts and damage they cause. So for the life cycle inventory on the left, there was already some research published a few years ago by Quantis, which is an environmental consulting company in Europe. And these are called the Plastic Leak Project Guidelines. Then they identified for microplastics, for example, tire abrasion particles or fibers from synthetic textiles, as well as plastic pellets during the plastic production. So what is the leakage of each of them? As well as also for macroplastics. What is plastic littering? How are microplastics leaking to the environment? And they look into different transfer pathways. So for example, for the microfibers, they looked into the wastewater pathway, or as well for the tire and rubber particles, for the road runoff or the air pathway, and for the microplastics uncollected waste. And ultimately, the plastics end up leaking into different environmental compartments, such as ocean, fresh water, soil, and air. And then with time, they're redistributed. For example, a river could transport plastic in a plastic bag all the way to the ocean. So the final release compartment in this case would be the ocean. So there's ways to quantify this, or at least have some good estimations. But what was lacking was how to connect these plastic emissions with the damage they actually cause on the environment. So this is why Marilka was founded, and Marilka is a scientific committee across the world. I'm part of this scientific committee. And in Marilka, we take the inventory, so the emissions of plastics of different sizes, macro, micro, and nano. We look into the fate in different environmental compartments. So for example, in the marine compartment, how long does it take for a plastic to degrade? This would be one of the fate mechanisms. Then we look into the exposure and the effects. So for example, on humans, the exposure could be inhalation, ingestion, and the effect would be human toxicity. And ultimately, what is the damage caused on human health? And this is a quite complex framework. For today, I will be focusing mostly on ecosystems, which is my research. But if you have questions about the other parts, you can ask me in the question period. So for the ecosystems, we at Marilka propose 3 different impact categories. The first one is ecotoxicity, which is linked with plastic additives, for example, that could desorb from the plastics once they're ingested in the animals and cause toxicity problems. Or also plastics in the environment could absorb certain chemicals or metals and transport them into certain organisms. So this is what we categorize as ecotoxicity. Then there's the physical effects on biota, which is currently the main focus of my research. And for the case of microplastics, this would be a physical abstraction, or just by the physical presence of the plastics. If they get ingested, they could accumulate in the body, for example, in the guts, leading to health issues. Or for microplastics, physical effects on biota could be entanglement, so that the plastic ends up wrapped around the animal, causing health issues. So there's just a physical presence separated from the toxic effects. And finally, we have invasive species, and research has shown that different species can attach to plastics and can get transported far away where they're non-native. This could be bacteria, viruses, or even bigger organisms. So these are the 3 main categories. And as I said, I mostly focus on physical effects on biota in my research, specifically for microplastics in aquatic environments. So how does this translate into modeling? Well, we need characterization factors which link the microplastic emissions in kilograms of plastic with the damage they cause on ecosystem quality and potential disappeared fraction of species over a specific area and time. And as a common structure in the methodology of LCA, we have different sub-factors for this characterization factor. So in this case, the fate factor is linked with the distribution and longevity of microplastics. The exposure factor is linked with the ingestion of microplastics. And finally, the effect factor is linked with animal health issues and reproductive impairment. And last year, my colleague Jérôme Lavoie at CEREC published a combined exposure and effect factor for nanoplastics and for microplastics for aquatic ecosystems. So he covered both freshwater ecosystems and marine ecosystems. And the way he did it is that he looked into different micro nanoplastic concentrations and looked for experimental studies of different species across 9 different phyla, and looked into what is the potentially affected fraction of species for a specific microplastic concentration. So with this, you could have a HC50, and from that you can extract based on LCA methodologies and exposure and effect factor. And in my work, I focus on the missing part, which we're missing to get the full characterization factor, which is the fate factor. So just a note that I have a colleague at CEREC who within a year will publish a very extensive fate model of how microplastics get transported in the marine environment. And so it's quite complex, but my work, which was before that, was more exploratory to see which are the main fate mechanisms and where we should focus our research efforts. So in this case, we looked into the marine environment. For the future, we will look into also the freshwater fate. And for the marine environment, the 3 fate mechanisms that I tested were fragmentation, so the breaking down of macroplastics into microplastics, degradation, which in Marilka we define as the quality loss of the plastics, so the breaking down of the polymer chains for example, by bacteria that could eat up the plastics and ultimately mineralize the plastics. And then sedimentation, which would remove the microplastics from the water column and transfer them to the sediments. So of course, microplastics could potentially have effects in the sediments too. But in this case, we're just looking for this particular research at the effects at the water column. And based on literature values, there's a high uncertainty. So I looked into literature and took the worst case scenario, average case scenario, all the way to best case scenario for these different fit mechanisms. So there is a detailed modeling behind, and we can go into the details if you're interested in the question period. But for now, I will show you the results for these characterization factors. So on the left, we have the results for tire and roadway particles. And on the X and Y axis, we see degradation and sedimentation for different cases, fast, medium, and slow. And we can see for tire and roadway particles, actually the degradation barely affects the results, whereas sedimentation does have a visible effect on the results. And this is because the tire and roadway particles are so heavy that they sediment very quickly. So there's not enough time for degradation to be relevant as the tire and roadway particles are in the marine water column. And differently to this, expanded polystyrene, so on the right, is very light, and it is expected to float unless there's biofouling, and it could become heavier and sink. So there's a high uncertainty associated to the sedimentation of expanded polystyrene, as well as uncertainty in the degradation of expanded polystyrene. So both at this point play a role in the characterization factor. And note that on the Z-axis, actually for expanded polystyrene, the characterization factor is several orders of magnitude higher compared to tire and roadway particles. So this means that the potential impacts of 1 kilogram of expanded polystyrene are on the ecosystem quality are higher, much higher than the one kilogram of tire and roadway particles. So in order to see how we can apply this to a case study and identify what are research shortcomings, we did a case study which was similar to the one I showed you earlier in the presentation, but now a few years later in a bit of a different scenario. So it was a case study which compared containers to carry a meal for one person in a food court in Montreal in 2021. And we compared 3 single-use containers, so 1 expanded polystyrene made in Ontario, and 2 compostable alternatives, 1 bagasse container made in China, and 1 wood pulp container made in Quebec. And we collaborated with this company in Quebec to give us the data. And then also we had a reusable ceramic plate made in China. And we looked into the environmental impacts across the entire life cycle. But for the sake of this presentation, I will look just into the plastic inventory, which is the focus of my research. But we also did the inventory for CO2, for water, and for all kinds of other emissions. So for the plastic inventory, we used the plastic leak project guidelines, and we identified for the different life cycle stages where were plastics potentially leaking to the environment. So that's the part of the production, the pellet losses as the plastics are produced. Then there's the transportation. So between each life cycle stage, there's transportation by trucks. So there can be tire abrasion and particles can be released. And finally, at the end of life, we have either direct littering on the street or landfill, which even if people dispose the container to garbage, maybe there could be some waste mismanagement, and this could lead to potential leakages to the environment. And note that in the Plastic Leak Project, they do give data for almost all countries in the world, because it varies a lot how the waste is managed in different countries, and this will lead to different leaks. So an open dumpster will lead to way more leaks than just a closed sanitary landfill in Canada. So in this case, we were taking the data for Canada specifically. And we can actually see that the leakage for transportation, tire and rubber particles, is several orders of magnitude smaller than for the other categories. Although note that the unit is different, so in this case, this is per kilometer of transport. But even if we multiply it by 1,000 or 10,000 kilometers, it's still smaller than the other life cycle stages, the leakage there. So now that we know what is the estimation of the plastic leaking, we can link that with the characterization factors to see what is the ultimate damage on ecosystem quality. So here are the results On the left, we see that the highest potential damage comes from bagasse, which is mainly linked with this category in orange, which is climate change. And this is mainly because of the electricity mix, which was used in the production of bagasse, which is mainly coal in China. So this is why there is high potential of climate change impacts. Then for the wood pulp container made locally here in Quebec, the potential are much smaller, and they would be comparable to the ones of the expanded polystyrene container if we did not take into account this part in gray. So the part in gray is the part linked to the plastic emissions. And we do have a high uncertainty. So this is why I put this table here. In the best case scenario, the results of the plastic physical effects on biota impact, they're so small that they're not visible. But actually in the worst case scenario, they would almost double or more than double the potential impacts of expanded polystyrene. So we can have a closer look into these impacts from microplastics. And we can see for the different life cycle stages. And at the bottom, we see there's different scenarios, best case, average case, worst case, but still this doesn't change the proportion much for the different life cycle stages. So we see actually potential impacts coming from transportation are very small. There's a few for production, and then the highest are linked with the end of life, particularly with direct littering. So if we were to reduce the littering, there we would have the biggest change in order to reduce these plastic emissions and the damage they cause on ecosystem quality. Now, we can also look into the human health results, which unfortunately in this case, we do not have characterization factors or methodologies for the impacts of microplastics on human health. But there's a research project starting now, so this is to come in the future. But I still wanted to show you for the other impact categories how it looked like. And again, we see that bagasse has the highest impacts overall on human health. And this is one more time linked with the energy mix, which was mostly coal, which is linked with climate change in orange and also particulate matter formation in yellow. So this is why overall bagasse has such high impacts compared to the other food containers. And then we see that the wood pulp container and expanded polystyrene container are close in the same order of magnitude, but we're lacking all the potential impacts from plastic emissions here. So this could change in the future. So note that what I showed you today was just for marine impacts of microplastics on physical effects on biota. And there's a lot of research ongoing, which in the future will contribute. So we're lacking the impacts of the plastic additives that are leaking, for example, invasive species, and all the whole framework that I showed you earlier on, even the macroplastic entanglement. We're just looking into the microplastics. So this was just a snapshot of the current research, but more will come, and this could potentially be adding more and more layers to this. And we don't know if ultimately the expanded polystyrene container would be worse than the bagasse or not. So we don't know at this point. And well, as a conclusion for the part of the methodology, we do need to continue the research and the detailed modeling of different types of microplastics to reduce uncertainty. And we did see that there was a big difference between the potential impacts from tire and rubber particles and expanded polystyrene. So this is quite polymer specific. And in my ongoing work, I'm developing characterization factors for 6 to 7 types of plastics, which should be published up to the summer. So this is ongoing work. And we also need research for different environmental compartments. We're just focusing mostly on the marine compartment, but also for the freshwater, we already have the effect factor, but we're lacking the fate factor, so should be coming in the future. And also different impact categories, as I mentioned. And for the case study, we saw that the compostable alternative may not always have low environmental impacts. So for the case of bagasse, it was much higher. For the case of the local wood pulp container, it was lower. So it's not a universal answer that just compostable is good, because it looks only at the end of life, but not into the whole life cycle. And then also we saw that in this case, the littering has a higher potential damage on the environment rather than the leakage from the tire and rubber particles. So with this, I finished my presentation, and I'll be happy to take any questions you may have en anglais ou en français. Thank you for listening.